Next is a very important 5-mark brief answer, and it is explain the process of formation of complex proteins. So over here individually we have to explain everything: transcription, translation as well as translocation, step by step, right? So let's have a look at the steps. So in the first point what we mention is that different proteins need to be synthesized, right, according to the instruction present on the DNA, and these proteins are very important for our body, right? So information about protein synthesis is stored in the DNA, as I told you, inside the nucleus which is present, right, and synthesis of the appropriate proteins as per the requirement is necessary for the body. Let's have a look at the word over here, appropriate. Appropriate proteins means the required proteins, the necessary proteins. Let's highlight the word, let's split it into parts so that we can understand it. We can split it in 3 parts, appropriate. Alright, now in the next point we are going to mention something about the central dogma, that the DNA will first be converted into mRNA, right, and then that mRNA goes into the cytoplasm, and with the help of that proteins are going to be made, right. So these proteins are synthesized by DNA through the RNA, and this is called as the central dogma of life. So a set of rules that are common for all living organisms, that is called as the central dogma of life. Let's have a look at this word over here. Synthesized means produced. Let's split it into parts so that we do not make spelling mistakes. Synthesized is how we can split it into parts. Then we talk specifically about this mRNA. It is produced as per the message of the nucleotides on the DNA. But in the next point we have to mention that both the strands of the DNA are not important for this process. Only one of the 2 strands of the DNA are important for this process. So only one of the 2 strands of DNA are used in this process. That is what we mentioned over here. And once that copying is done, the mRNA is produced, and this is called as transcription, because a transcript, a message has been produced over here. So the sequence of the nucleotides on the mRNA are exactly complementary to the sequence of the nucleotides on the DNA. Right, it is going to be not be the same, it is going to be complementary, in the sense if the DNA has guanine, the RNA is going to have cytosine, right. If the DNA will have adenine, the RNA will not have thymine, instead of that it will have uracil. This is what we call as the complementary sequences, and this is exactly what happens in transcription. So that is what we mention over here. So this process of RNA synthesis is called as transcription. Now that we have mentioned about transcription, let's mention about translation. But for that, the mRNA has to move from the nucleus into the cytoplasm via a nuclear pore, right. So we mentioned about that. The mRNA formed in the nucleus comes into the cytoplasm. Once in the cytoplasm, a ribosome will attach to it, and it will start translating it. So it brings the coded message from the DNA. We already know the mRNA is complementary to one of the strands of DNA, so it has brought that message along with it over here in the cytoplasm. The message contains the codes for amino acids. It is very important whenever the ribosomes are going to translate the mRNA, what are they going to translate it into? They are going to translate it into a bunch of different amino acids, right. The code for each amino acid consists of 3 nucleotides. Each amino acid will be coded by a set of 3 nucleotides. Together we call them triplet codon, right. So it is called as the triplet codon. That's what we mentioned over here, right. Next we talk about the mRNA being made up of thousands of triplet codons, because the protein that is going to be synthesized, it itself might be made up of thousands of amino acid, or it is yet to be made up of thousands of amino acid. So if 1000 amino acids are present, to code them, obviously thousands of triplet codons have to be present, right. So that is exactly what we have mentioned in the previous point. So as per the message on the mRNA, amino acids are supplied by the tRNA, very important, transfer RNA, so which has something called as the anticodon, and which is complementary to the codon. The name itself says it's anticodon, and because of this codon anticodon complementary sequences, the correct amino acid is brought at the correct location in the ribosome. So for this purpose, the tRNA, as I said, has anticodon having complementary sequences to the codon of the mRNA. So they bind together, and once 2 amino acids are brought close to each other, there is another RNA called as the rRNA, ribosomal RNA, that creates peptide bond between these 2 amino acids. So this process, first of all bringing them, is called as translation, right. Now we talk about the amino acids brought in by the tRNA are bonded together by peptide bond. A very, very strong covalent bond is the peptide bond with the help of ribosomal RNA or rRNA. Now, for complete translation, obviously the ribosome will have to move from one end of the mRNA to another step by step, right? So, it is as if it is changing its location, and that's why it is called as translocation. That is the last part of our answer that we explained over here. During this process, the ribosomes keep on moving from one end of the mRNA to the other end by the distance of one triplet codon. Not a lot, one triplet codon at a time, so that there is enough time given for the tRNA with its anticodon to come and attach to the codon of the mRNA. And this is called as translocation, the changing of the location of the ribosomes on the mRNA. Right, and this is exactly how complex proteins are made. So many such chains of the polypeptides, in fact, they come together, they bind together, they are twisted and turned in the right manner, they are folded, and that makes our complex proteins. So this is the process of the formation of complex proteins.